Howdy, this is Lab Report 9, Phage. Group 2, Ana Martinez, and
And Hannah Garcia, Microbiology Fall 2024.
Introduction and Significance. The purpose of this lab is to quantify the concentration of phages in a sample by measuring their ability to infect and lyse bacterial cells. They form clear zones called plaques on a bacterial lawn. This lab helps determine the concentration of viable phages in a solution, phage titer, which is important when experiments require a precise amount of phages. PFU phage labs are crucial for accurately measuring phage infectivity, observing phage bacteria interactions, and understanding phages for effective research in clinical and ecological settings, such as effectiveness of phages against antibiotic resistant bacteria. The materials: E. coli, 10 to the power of negative 1 dilution of phage, T4R in a microcentrifuge tube, 4 LB plates, 9 microcentrifuge tubes, sterile water, micropipette and tips, 4 large test tubes. The methods: make a serial dilution of phage on sterile water. Obtain the 10 to the negative 1 dilution. Obtain 5 microcentrifuge tubes and label them 10 to the negative 2, 10 to the negative 3, 10 to the negative 4, 10 to the negative 5, and 10 to the negative 6. To your new tubes, add 900 microliters of sterile water to each. From the 10 to the negative 1 tube, obtain 100 microliters and add them to your 10 to the negative 2 tube. Mix well. From the 10 to the negative 2 tube, obtain 100 microliters and add them to your 10 to the negative 3 tube. Mix well. Repeat these steps until you reach tube 10 to the negative 6. Once you reach tube 10 to the negative 6, discard the extra 100 microliters. If we were to continue the serial dilution, this is what we would add to 10 to the negative 7. Whole plate titter from serial dilution. Prepare a 1 LB plate by labeling it on the agar side. Include the date, type of phage used, dilution used. 10 to the negative 3, 10 to the negative 4, 10 to the negative 5, or 10 to the negative 6. Obtain 4 microcentrifuge tubes and label them E negative 3, E negative 4, E negative 5, and E negative 6. In this case, the E will stand for E. coli and the negative numbers will stand for the power of dilution. From your original serial dilution tubes 10 to the negative 3, 10 to the negative 4, 10 to the negative 5, and 10 to the negative 6, move 100 microliters of the dilution into the new E-labeled tubes. Move 100 microliters of the E. coli stock culture into each microcentrifuge tube containing phage dilution. You now have 200 microliters in the tube, 100 microliter phage, and 100 microliter E. coli. Vortex the tube briefly to mix. Allow the tube to sit on the bench top for 10 minutes. Make your way over to the water bath with the following items: 1 LB agar plate that you labeled, the dilution microcentrifuge tube with E. coli and phage, which label matches that of the microcentrifuge tube. The following steps must be done quickly, so read carefully before you start. Obtain 4 large test tubes. Pipette the phage bacteria mix 200 microliters from its microcentrifuge tube into the top agar tube, 5 milliliters. One at a time, add 5 milliliters of molten top agar from the water bath to a large test tube and immediately pour the contents onto its respective LB plate. Swirl gently about 3 times to distribute the molten agar across the plate before it solidifies. Replace the lid and allow it to sit undisturbed until the lawn sets, 5 to 10 minutes. Keep the agar side on the table. If you need to move your plate, keep it as level as possible. Invert to incubate per film and place in your basket and return to the incubator. Results and observation. The following slides are the results and our observations of the incubated phages on the LB plates. The pictures are our results and the observations are based on what we recorded on our lab notebooks.
Results and observations. All plates had too many plaques to count. The overlapping of bacterial lysis by the phages made by the plaques, clear zone, impossible to count. However, the amount of plaques did seem to decrease as the phage concentration was diluted. Calculating PFU. Number of plaques over dilution factor times volume of diluted virus added, or PFU by milliliters. Number of plaques 175, dilution factor 10 to the negative 5, volume of diluted virus added 200 microliters or 0.2 milliliters. 175 plaques over 10 to the negative 5 times 0.2 milliliters equals 8.75 times 10 to the 7th PFUs per milliliter, or 87,500,000 PFUs per milliliter. Conclusion. Some possible errors in this lab that prevented being able to count the number of plaques could have been that the dilution factor was too high, errors in diluting or mixing, contamination of phages during the process, and uneven distribution of phages, which lead to having a high amount of plaques present on the LB plates. Thank you.